Okay, well, we'll go ahead and start. My name is Amanda Dumas. I'm an associate professor of clinical pediatrics, and I'm a former Roadmap scholar. And so I just want to welcome you all today to those who've joined our seminar series. The Louisiana Center for Translational Sciences, CaTS Center, is proud of Louisiana's scientific response to the COVID-19 pandemic. And researchers from around the state are working on the front lines, conducting research to improve testing, surveillance, health impacts, and treatment. And as a resource to all scientists, clinicians, and citizens, we've launched this Louisiana COVID-19 Chronicle Seminar series. It was started in October of 2020, and the LA CaTS Center is hosting a virtual reoccurring COVID seminar series featuring scientists and clinicians to showcase the work being done right here in Louisiana in the battle against COVID-19. Topics and speakers for each seminar are announced to the Louisiana CaTS email list, a calendar of events page, and Twitter as they're finalized. We want to congratulate the researchers who've been successful in contributing to our knowledge on COVID-19 and hope you will join us for future episodes of the Center's Louisiana COVID-19 Chronicle Seminar series. So during the talks, please type your questions in the chat window. And we will save time at the end to share your questions with each speaker, either from the chat or you can unmute yourself at that time. And so first I'd like to introduce Dr. Chelsea Kracht. She's our first speaker. Dr. Kracht is an NIH T32 postdoctoral fellow at Pennington Biomedical Research Center in Baton Rouge, serving in the Pediatric Obesity and Health Behavior Laboratory. Her research focuses on optimizing child health before age 6 years, including environmental influences on child movement and behaviors, such as physical activity, sleep, and screen time. She will present her work on household chaos, routines, and family movement behaviors during the COVID-19 outbreak. So thank you so much. And if you'd like to go ahead and share your screen, we'll be all ears.
Great. Well, thank you again for having me. Sorry, I had a little household chaos right there. And thanks again for everyone joining. Again, I'll be sharing on some work I did during the COVID-19 outbreak. So as a part of today, I'm actually going to be, presenting two results from the same survey. So we're going to be talking about the child results and the maternal results. I do want to be upfront that the child results are currently unpublished. They are under review. The maternal results were just accepted recently to a journal, so these will be published shortly. Again, both of these focus on household chaos and movement behaviors and take just kind of two different lenses to look at how it's influenced both child and mother health behaviors. So today I'm going to start here with the child results. So you may have heard about the recent movement to look at physical activity, sedentary behavior, and sleep, but I bet you haven't heard about the 24-hour movement guidelines. These guidelines are a huge step forward for physical activity and public health because they finally consider all three behaviors within the 24-hour day. Now, these behavior guidelines were actually started, or the conversation was started by a Canadian group about 5 years ago, but it wasn't adopted by the World Health Organization until 2019. And if you've been keeping up with any of this movement behavior literature, you know that the World Health Organization just created physical activity and sedentary behavior guidelines for adults and children just a couple months ago. Even though these guidelines are still relatively new, what we do have is some literature that's been coming out in the past couple of years. And what's really alarming is that even at this young age range, very few children are meeting all three guidelines. So 5 to 15%. This is pretty concerning, especially in this young age range, because we know meeting more guidelines associated, especially with better cognitive development. And for obesity, the results are kind of mixed right now, and that likely has to do with the adiposity rebound that occurs between 3 and 5. But in order to really address all three behaviors, you really need to think about where all three behaviors occur. And in this case, especially for young children, that would be the home. And within the recent home literature, there's been this new term, or well, maybe picking up more speed, new term of household chaos. So household chaos has been emerging as a risk factor for obesity, but to be fair, it's been in the literature for about 20 to 25 years, predominantly looking at child cognitive development. Now, you may have your own definition of household chaos. It is the COVID-19 pandemic, but in the literature, we look at three separate pillars. This includes disorder, crowding, and noise. Disorder may be seen as are things where you expect them to be, do things happen on time, are things in general planned. Crowding can be seen not necessarily as having more people, even though it does correlate well with having more people in the home, but are there overlapping schedules? Can you do something within your home without kind of running into someone else or running into someone else's activities? And then noise can be seen as environmental noise, but if we look in the literature, noise is usually associated with screens and TVs being on. So this is more, it's making it difficult for it to have like a calm and quiet environment to relax. As I've described household chaos, it may be no surprise that household chaos is associated with more screen time and less child sleep. Now, there isn't any household chaos interventions per se, but there are interventions that have facilitated less screen time and more sleep that have focused on routines, which would really address this disorder component of household chaos. So it could be said that routines may dampen or hopefully prevent some of the detrimental effects of household chaos. Now, as I'm presenting this, I guess, research study, if it was about a year or so ago, I would have told you a very different slide deck, but we had this question around February 2020. And unfortunately, we know a lot of different things occurred in March 2020, including the COVID-19 outbreak. Now, when we first had the outbreak, I'm sure there's a lot of things happening and a lot of literature kind of just popping up. And there's especially some literature coming out that there were compromised child movement behaviors in school-age children. And so we had reports that older children were sleeping more, had more screen time, and less physical activity. And I saw those reports really early on, and I said, I don't know if that really would apply to a preschool age range, because a lot of these changes with sleep are likely anchored towards a school start time in the older age range and the eveningness tendency. So in the younger age range, we have nap and we have a couple other things, and they are going to be more influenced by the parents at this age. And so I wasn't really sure. I looked at those results and it kind of made me think, what would be happening in young children? But also, let's just say, I think many of us had anecdotal evidence that it was a pretty high chaos time. So we felt, we had this idea going forward, and this would be an optimal or maybe unique environment to look at the relationship between household chaos and child movement behaviors. And really, when we were looking at household chaos, we weren't just looking at, routines as an opportunity to facilitate sleep, screen time, physical activity. We thought that, during this time, this disruption and what behaviors are disrupted might help us understand how all these behaviors work together, which comes to our proposed model. So our proposed model is disruption in the home could help us understand all these movement behaviors. So we have independent variable household chaos to our dependent variable of sleep, screen time, and physical activity. Now, we felt it wasn't enough just to look at independent variable and dependent variable, but what are we going to tell families and what are we going to tell those taking care of children at this time? And so we said, what if we could look at routines? So if those who had routine during this time had better sleep, had less screen time, and more physical activity. So we also looked at the role of routines within this study as something that could kind of reset household chaos, which led us to the CHILCO study. So as you may notice, it was chill, and then we added the CO because of COVID-19. And we had two aims here. So we were first looking at household chaos and movement behaviors. Our big thing that we were looking at here was physical activity. We hadn't really seen if physical activity was going to be influenced by household chaos or related to. And then our aim two was to look within these household chaos categories and see if routines were related to better amounts of health behaviors. So the CHILCO survey was an online survey for mothers in the US of preschool age, that's 3 to 5.9, so right before kindergarten entry. And this happened around the stay-at-home order phase 1 for most states. We distributed it primarily through Facebook and other social media, and we used validated questionnaires that I'll talk a little bit more about in a sec, and looked at other report of changes in movement behaviors and barriers to routine. Now, I say all this on the slide, but it was a really crazy time. Oh, pardon, went a little too fast there, but our protocol was submitted March 13th. As I mentioned before, this was a question we had been looking at before. So once the COVID-19 pandemic hit, I already had the protocol, and I had a meeting with me and my mentors, Amanda and Peter, and I was like, we can't wait on this. And so we had our protocol together pretty quickly. We had our virtual startup meeting, and our survey went live May 1st through 24th. We got our first paper out pretty quickly right after that. And a lot of people moved really fast for us to do this, so I wanted to really thank them for moving so quickly at a crazy time. And with that, and with this survey, again, this survey was already kind of constructed, so we already were going to use the ChaOS scale. The ChaOS scale focuses on daily disruption. And I really want to make a point here because this is very different than adverse childhood events such as ACEs, so a divorce or moving. This is kind of the daily disruption within your home. And we looked at specific categories based on previous literature. For child movement behaviors, we use the NHANES questions, which focus on number of days you meet the moderate to vigorous physical activity guideline, light physical activity guideline, and then hours a day for sleep and screen time. We looked at various routines that were specific to our movement behaviors, so bedtimes, and then the importance of family routines, and then use linear regression with adjustment for covariates and other movement behaviors. So, So for our participants, we had about 1,800 included in analysis. We were aiming for around 1,600, so we got for completion, so we got right around that number. Within our sample, compared to previous pre-pandemic samples, we did notice that many households were classified as moderate to high and high. This sample was predominantly white and middle income, which may not be generalizable to others, but I would say, especially at this time of the pandemic, these were people who were definitely getting hit by a lot of changes. So to go to our first really look at movement behaviors across these CaTS categories, I have this figure here, and you may see right away here on the top, it's a pretty clear dose response. So we have meeting the light physical activity guideline here on the left. And on our kind of our bottom axis, we have low to high CaTS categories. We see a pretty clear dose response. Those in the low CaTS categories had more days of meeting the MVPA and light physical activity guideline compared to the high CaTS categories. And then similar for the sleep in hours per day, where there was less sleep in high CaTS and more screen time in high CaTS. I did want to point out that, we kind of saw about a third for physical activity. So no real huge decreases or increases, but many children slept the same, and there were more screen time, as you may have guessed as well. As for our actual AIM-1, I have a lot going on here, and I'll just hit the high points. But to orient you, here on the left, we have household CaTS score, and we also have the categories I mentioned before. And our low CaTS is the referent. For family routines, we looked at bedtime ritual, a consistent bedtime. And for us, I think it was important to really think, is it what's happening now, so routines since the outbreak, or is this some sort of learned trait? So having routines before was actually more important. So we wanted to look at both of those. As for our actual dependent variables, you'll see here with light physical activity and moderate to vigorous, household CaTS was negatively associated with days meeting the light physical activity guideline, MVPA guideline, but this was really predominantly in the high CaTS households. And then very few family routines were associated with physical activity. Oh, sorry. And as for sleep and screen time, this was exactly what we expected. So more household CaTS was associated with less sleep and more screen time. And you can even see there's a kind of a direct trade-off here with more routines associated with more sleep and less screen time. So if I were to sum up this table, I would say household CaTS is related to less physical activity, less sleep, and more screen time. Now, looking at our kind of our AIM-2, so looking within the CaTS categories, I mentioned before that we were aiming for a sample around 1,600, and the goal was to really conduct this analysis. So we have quite a few in each of these categories so that we could look within categories and see some of these relationships. So I have low, moderate, high, and high CaTS categories here. So this is kind of separate linear regression models. And then we have our outcomes right here. And I don't want to get too much into the weeds here, but I will point out that in general, a bedtime ritual was associated with more sleep. This was not apparent in the high CaTS household, so may need to look a little bit further into those. And then there are various things associated with less or more screen time. Now, you may notice there's a column missing here, and for good reason, because no routine was associated with physical activity within these CaTS categories. So we can't say routines is associated with more physical activity regardless of your CaTS category. So bringing this all together, we for AIM-2, I'd say amongst differing household CaTS groups, no routine metric was related to additional days of MVPA. And we do have some individual considerations for sleep and screen time within these other CaTS categories. So overall, we did see that there is a high household CaTS, and we didn't see a change in sleep like older children. That's probably due to the difference between having a school start time. I will say that we did see limited associations between sleep and, routines and sleep in high CaTS households. And I want to really make a point of that because one of the big things with chaos and sleep is that's really the main association that's been looked at over the last couple of years. And so we really need to look beyond routines, looking at those high chaos households. Now, recent literature has come up since we've conducted this study, and there was an intervention that came that was published around June or July that was a routine-based intervention and did measure chaos before and after. And they did find a decrease in household chaos, and that was related to child sleep. So kind of even tying all this together, there's clearly something going on with chaos and child sleep and screen time. And the lack of association between kind of these routines and physical activity is actually shown in some of our other work, other Pennington collaborators, that household organization was associated with sleep and screen time, but not physical activity. So, there's a lot of things to consider with this sample. It was large and we did get it at a critical time. And we, had a lot of these measures ready to go, which is good because as the chaos literature has evolved, people have used very different definitions, and we've used one that has been a part of the literature for the last 30 years. But I do recognize that the household chaos scale is a scale, and that's exactly what it is. There's no clinically meaningful cut points, such as like when in the Pittsburgh Sleep Questionnaire, there's a cut point of, sleep disruption, but here it's just kind of a scale. So above 50 doesn't mean that you're at risk for obesity or anything like that. This is also a predominantly high income sample, and we only measured once. So I would say if we're going to move to the next step, this household chaos and physical activity relationship needs to be explored a little bit more. And I think it means looking beyond routine, looking at those other two pillars. We also need real-time measurements, so ecological momentary assessment, and also evaluation of these in diverse and low-income samples and serial measurements. So is household chaos always high, or are there times when it's lower? How does that kind of work together? And of course, support all families to promote routine for healthy development and just overall health for the family. Now, again, that was my first, that was the main purpose of the Chilco study. I'm going to transition to the mother results. I'm not going to go as deep into these, but because we have a lot of the similar methods, but this is a very different look at what household chaos is doing within the home. So as you may have guessed, household chaos associated with lower maternal sleep and obesity. But what I bet you don't know is that mothers of young children are at an increased risk for low levels of physical activity and sleep compared to mothers without children and mothers with older children. So this is really a special group that gets really impacted by their caregiving and many hats that they wear. But the thing that always comes up when we look at these maternal health behaviors is this household chaos or is this stress? And I think that's a good question because a lot of people actually classify household chaos as a form of stress, even though it's daily disruption. So it's kind of a different mechanism. And so for us, one of the questions was, well, would stress or household chaos, or maybe combination of both, be related to changes in mothers' physical activity and sleep? And really, what are some things that we could recommend for routine? Especially during the pandemic, mothers are disproportionately affected by job loss, household demands, and care for young children. So what can we suggest to promote routine? Which led us to our maternal paper or maternal results, which were to investigate the relationship between household chaos, maternal stress, and maternal physical activity and sleep, and then look at barriers to home organization. So we use similar methods. This is the same survey. The mothers did report their own stress, health behaviors, and barriers to home organization. For AIM-1, we used mediation analysis using process, and then for AIM-2, we did linear regression and frequencies. We adjusted for many of the same variables that we did in the previous publication. So what did we find? So about half met the physical activity guideline, half met the sleep guideline. So, initially this doesn't sound too bad. And, I'm getting a little bit into the discussion ahead here, but I did want to put it in perspective to the previous publication. So we know pre-pandemic, mothers of young children, the range is between 35 to 45. So this result, maybe it's a little bit higher, but not too concerning. Now for mothers of young children, previous literature doesn't look at meeting the sleep guideline. They actually look at how much short sleep you have. So in our sample, we had about 50.3% had short sleep. So you may have guessed no mom reported more than 9 hours of sleep. And pre-pandemic reports find that 25% of mothers of young children have short sleep. So we have twice as many mothers with short sleep. And this was a couple months into the pandemic. So thinking about the relationship between household chaos and stress and all these health behaviors, In the mediation analyses, we found that household chaos was associated with stress, and stress was negatively associated with these health behaviors. And household chaos was then negatively associated with health behaviors. So in this full mediation model, though, we found that stress only partially explained the relationship between household chaos and maternal health behaviors. So stress was not the reason, did not explain all of what household chaos was doing. So these are independent factors. Now for AIM-2, we wanted to get a little bit more into what's really going on within the household. And so the COVID-19 factors that were significantly associated with household chaos included decreased income, lack of non-parental care, and teleworking. I want to say that a second time. So mothers who are teleworking, either part-time or full-time, reported more household chaos compared to those who are not teleworking. And of course, very concerned for the virus diagnosis. Accordingly, the barriers to home organization really mirrored what we found with the COVID-19 factors, including lack of childcare, changing job schedule, and lack of family support. So bringing all this together with the maternal results, we actually did find similar results to other parents of older children within the pandemic literature, if you may, but twice as many mothers reported short sleep, which is pretty concerning. A couple months into the pandemic, and it's likely that this has persisted for many months, and just the detrimental effects of short sleep on, physiological and mental benefits as well. I will say that just like taking a step back from pandemic literature, getting this literature published was very interesting process, but I'm actually kind of glad that this, it took us a couple of journals to get this published, because when we originally chose these factors related to home organization, we based it off of some COVID-19 literature at the time, but the more literature that came out that really described the mother's role during the COVID-19 pandemic, including the increased focus on childcare, the trying to fit work in as they could, and the increased housework, really put it together as to how all this might contribute to household chaos. And so we found that the corresponding factors related to childcare work, and I say work with an asterisk if they haven't lost their job or decide to leave the workforce to take care of young children, and housework were associated with more household chaos. This really goes to what our conclusions are for the next steps. And the big conclusion was, household chaos is independent beyond stress for maternal health behaviors and should be considered as something to be addressed in these future interventions. And really to promote routine and maternal health, safe and affordable non-parental care options. There's not only center-based care, but grandparents at this time may be limited. With more vaccine. Hopefully that's becoming more of an option for mothers. Supporting mothers in the workforce, because as other literature has shown, mothers are losing their jobs, but here we found a decreased income was related to more household chaos. And then of course, we're always chasing the endorse a healthy work-life balance. And that really includes, there's so many factors to go into that, but I don't know how to answer that question, but that's what we really need to focus on, is the work-life balance and, some way for mothers to get some sort of a break. But again, I just kind of talked about what I did over the COVID-19 pandemic, but a lot of people really put a lot of work into this, including my mentors who serve as co-authors. And the most important people to thank, of course, are the mothers who shared their experiences and participated. That's all I have. I welcome your questions.
Thank you so much. That was really amazing. There is one question in the chat from Dr. Van Dyke asking, I assume sleep was reported by the parents. Have you considered physiologic devices to measure sleep?
I think that's a great point. Yes, I think that's a great idea. We do have a collaborator who actually used wrist warning accelerometry during the COVID-19 outbreak. And I don't know what she found about changes in child sleep, but I can definitely follow up with her, but that's a great point.
And I was wondering regarding the child study as well, and I'm sorry if I missed this, but if siblings were both being measured, did you see differences between sibling measurements from the same household with the same measure of chaos?
That's a great question. So we asked mothers to, if you had 2 children within the age range, to only report 1, but we did find that having more children in the younger age ranges was associated with more household chaos. So for instance, if you had a 5-year-old and like a 3-year-old, you reported more household chaos. But once those who had children above the age of 10, that relationship wasn't there. So it's really having more younger children within the household reported more household chaos. But we actually didn't get them to kind of report on 2 children, just only 1 within the age range.
Thanks. Does anyone else have a question? You're welcome to chime in. If not, I do have one more related to the children again. In terms of the screen time, were you measuring the different types of screens being used? You know, for example, a parent's smartphone versus the television, which, you know, is loud and the whole house can hear, or you know, a tablet or something like that.
That's a good question. We did not. We use I believe NHANES includes TV, computer, tablet, smartphone. I want to say that's the big four. But that is a good question to really look at the context of this screen time. Is it TV or is it smartphone? And I know there's, you know, anecdotal evidence of people giving their kid more tablet time. So that's a good question. We didn't measure it, but that's something we should definitely consider.
Well, thank you so much. This was a really interesting presentation. Thanks for sharing. And again, this is recorded so people can access it or share it with their colleagues. All right, well, next we'll have Dr. Elizabeth Norton present. Dr. Norton is an assistant professor at Tulane University School of Medicine in the Department of Microbiology and Immunology. Dr. Norton's research focus is protective host immunity, and in particular, she's interested in how infection or vaccination can induce protective T cell and B cell memory responses in systemic or mucosal tissue compartments. Additionally, which factors from the host or which strategies during vaccination, like adjuvants or delivery systems, drive these protective responses. Dr. Norton will present her work on cellular immune response to SARS-CoV-2. Thank you so much.
Thank you for having me. Can everybody hear me okay and see my slides?
Looks good. Okay,
great. So I'm at the downtown campus of Tulane University, and I want to talk about some of the studies that we're doing with SARS-CoV-2 immunity. And if you think about SARS-CoV-2, obviously we've heard a lot about it, but you know, it enters the host, it binds to the ACE2 receptor, your immune response, and then it starts replicating, and then your immune response starts seeing those viral antigens and responding to it. So as kind of depicted on the right of my screen, the virus will be ingested by an antigen presenting cell. It'll get chopped up into smaller peptides to present to the appropriate CD4 T cell or CD8 T cell that is specific for that antigen. And B cells will also undergo clonal expansion that are specific for the antigen. In this case, B cells will see whole antigen, whole proteins, not peptides. Well, there's a lot of conversation about seropositivity, antibody neutralization, responses of antibodies, but equally important to the B cell response is this CD4 T cell response and this cytotoxic or CD8 T cell response. So the CD4 T cells will help interact with the B cells to make these antiviral antibodies and change even the functionality of those antibodies. Whereas, you know, the cytotoxic or CD8 T cells are known for directly destroying infected cells. And just to give you a little bit more detail, I know not everybody on this talk today is an immunologist, but when we say things like Th1, Th2, Th17, or cytotoxic lymphocyte, what we're referring to is these kind of programming of T cells to express certain transcription factors, certain cell receptors, and also what I'm going to end up talking about today is different cytokines. So a Th1 response, those cells produce a lot of interleukin 1 or IL-2, interleukin 2 or IL-2 and interferon gamma. And this plays a role in recruitment and activation of macrophages and dendritic cells. It also can, in particular interferon gamma, you know, produced by CD4 T cells or CD8 T cells, will have a direct effect on kind of viral infected cells or warning tissues that there's a viral infection nearby. But, you know, there are also other cytokines that can play a role, either positive or negative for the host, such things like IL-5 or IL-6 or TNF-alpha. So what happened during, I guess, the the last year's pandemic is that a group of investigators that included myself, we started wondering about the immune response in individuals who were being infected. And this led to a recent funding by the NIH for the Tulane University COVID Antibody and Immunity Network. And I am conducting studies under Project 2 on cellular immunity and memory to the virus. And the goal of this study is really to look at kids, adults with cancer, even maybe pregnant moms and cord blood or asthmatic kids, as many populations as we can actually find. And if you're actually have a population and want to collaborate, we would love to kind of test immunity in these different patient populations. But we take their plasma and their PBMCs, and then we're kind of looking post-infection at what happens to the immune response. Along with this project, we are also doing some mouse studies, but I'm not going to talk about that here. And our goal with the kind of our focus of cellular immunity is really to look at how does this change post-infection, and now, of course, with the vaccine rollout, post-vaccination. And you know, we know that this is going to peak, and we think that the T cell memory might actually outlast the serum antibody response. But more importantly, since this is the first time that there's been such a widespread coronavirus kind of pandemic, it's unclear how this kind of memory is going to be significant to health outcomes as kind of the virus circulates. Is it going to be like what happened with the first SARS, or is it going to be more like kind of the endemic coronaviruses that children get? So, and the reason why we came up with this initial hypothesis is because in investigators who had looked at individuals recovering from the first SARS outbreak, they did a 6-year follow-up study, and I'm publishing one of the studies on this, and they show that the anti-SARS IgG antibody titers, this is to whole virus, not any one particular protein, but these wane over time in the months after infection, whereas the interferon gamma response, you can see right now at the same time point, this last time point is still remaining highly significant compared to uninfected individuals. So this is why I think continuing to study the cellular immune response is going to be very important for SARS-CoV-2 and for COVID, understanding COVID disease. A number of recent studies have come out looking at what are the targets of T cell responses. Are we finding these T cell immunity in people who have been infected or hospitalized or whatnot? And a lot of these show a convergence that, you know, most of the CD4 T cell responses to the spike protein, which I kind of diagrammed here with an S, the nucleocapsid protein, or the matrix membrane protein. And, you know, there's different reactivity depending on the study and the people who are included in that study. But another important consideration that's come up is that there is some background that's quantified as like 50 to 60% of unexposed individuals have some background level reactivity in their T cells to coronavirus antigens and peptides. And that's thought to be because of circulating endemic coronaviruses. So the study that we started back in June and July, actually before this project got funded, was to look at people who were getting SARS-CoV-2 infected and coming down with COVID in the community, but didn't require hospitalization. So these subjects were recruited after their infection had cleared and their symptoms had resolved, or they were recruited from close contacts of people who had been infected. The age range was 1 to 80 years of age. And as I said, the study began in June 2020, which means that some of our individuals, we didn't have excellent kind of confirmed COVID tests for infection. Some we do, some have PCR results, but some we don't because they were infected before that was widespread. And these studies have, you know, really been led in the clinic by Monica Dietrich, Crystal Zhang, and then Kevin Susderick, Stacy Drury, and my lab have been all supporting all of this work in processing samples and whatnot. So what we do with the samples is when we collect that blood sample, we isolate and store PBMCs in plasma. And then we take the PBMCs to look at cellular immunity, and we do a 24-hour culture with antigens. And as I said before, we're interested in the S protein or the N protein or M proteins. And we use peptides when we can find them, because this way, these are peptide pools for those proteins that we know, at least in the early reports, most people are generating a good cellular immune response to. And we've been doing 2 assays that I'll talk to you about today. One is an activation marker assay, where you look at these CD4 or CD8 cells who have been stimulated for 24 hours, and you just see if they have an activation marker that's upregulated on their surface. And the other assay we do is just a recall cytokine secretion assay. So you just measure the cytokine response in those supernatants, culture supernatants. And as I said, we're interested in antiviral or Th1 immunity. And for those type of markers, we're looking at IL-2, interferon gamma, and TNF alpha in this recall cytokine response. But we also have measured different analytes such as IL-5, IL-6, IL-17, IL-10. And what we're thinking is that over time, as we get more and more populations, we'll be able to understand more of the relative contribution of perhaps this type of immune response versus people who have kind of a combination of like a Th1 biased immune response, or a Th1 and Th2, or balance with a T regulatory response. So what I'm going to go through first is just our activation induced marker analysis. So this is an example data of what we get. So this is the scatter of the lymphocytes that we acquire from the PBMCs that have been thawed. They get stained for CD3, that's a lineage marker for T cells, and then subsets the CD4 and the CD8. And because of an early publication on this, we also looked at a subset of CD4 T cells, which is CXCR5 positive, which is indicative of a T follicular helper cell. So these cells are known for stimulating B cells in the germinal center and being important for generation of a potent antibody response. And then once we have kind of our lineage markers and our populations gated, then we go ahead and look for activation changes. And this is these markers. We've used OX40, CD137, and CD69. OX40 is our main marker. And the type of data that we'll get is shown right here, where the change in expression. So mock will have kind of variable expression depending on the subject, but then if we stimulate those cells with the S protein, the spike protein, or peptides from the spike N and M proteins, we'll see an increase. And we report this as kind of a expression less the background, or the mock. And then what we're trying to do with this data is stratify it based on IgG seropositivity or IgG titers, COVID history, and demographics. And all of the data I'm showing you is kind of in process, because we are kind of getting everything together from all the people we collected before the vaccine rolled out. And we're hoping to get our final analyses finished by the end of probably next month to put into a publication. So if we look for activation induced markers for CD4 T cells, the CD4 subset that I said was very helpful for promoting B cell responses, the T follicular helper cells, or CD8, you can see very clearly that this OX40 activation marker really shows up in the CD4 subset, but not in the CD8 T cell compartment. So we've looked at a few. We looked at CD137 for our CD8 T cell activation, and that doesn't seem to be working very well, but we think the CD69 markers is gonna hopefully be a better indicator of CD8 T cell activation. And we really want to capture these cells, because these are the cells that can directly destroy virally infected cells. And so they're known to be important for viral infections. So this is kind of new data, because we've had a little trouble, I guess, figuring out a way to show activation of our CD8 cells. So for the rest of the talk, I'm really just going to focus on CD4 cells or just general cytokine secretion, which could be from either cell subset. So for T cell activation using this OX40 marker and just our CD4 population, if you look at subjects who were either seronegative, which is in the black circles, or seropositive, this is from one of our experiments of about, I guess, 16 people, you can see a clear enhancement of activation in the presence of the S protein that is partially recapitulated by using the peptides, not as strong as using the protein by itself. And if you try then to correlate this cellular activation with the anti-S titers, so presence of antibodies in these people. So here's our negative population. Here's people who actually generated antibodies and the level of these antibodies. What you can see is that this is not a clear linear correlation. In fact, there's a very modest correlation with our S protein and nothing with any of our peptides. The R value is not close to 1 or -1. So it's CD4 activation, at least by this assay, doesn't seem to tell us a lot about the magnitude of the antibody levels. And then, but what about those T follicular helper cells? So just so you can kind of see this, is that you have our CD4 T cell, and that these cells that are expressing CXCR5, well, that allows them to specifically home to the center of secondary lymphoid organs where they see their cognate B cell to provide this T cell help in the form of cytokines to this B cell that really help promote excellent kind of antibody secreting plasma cells or memory cells. So we would think that T follicular helper cells, at least the activation of those, might be indicative of a really strong antibody response. But actually we also see not a very strong correlation. I mean, it's trending towards an important relationship as we get probably more numbers, but in terms of like a linear relationship between the level of activation and our antibody response, that doesn't seem to be there. And then just looking at whether this memory is related to the day since COVID symptoms started in those subjects, or whether they had a fever, or whether they lost smell during the infection. None of this seems to be highly related, at least at this point, to our ability to detect activation in these CD4 cells. So now I'm going to move on to our responses of just looking at cytokine secretion. This is a more classic, I would say, assay for looking at immune responses to any type of infection or vaccination. It's just not as specific because we can't tell which cell is secreting the cytokines. So here's some data from a single experiment looking at secretion of IL-2, interferon gamma, TNF-alpha. These, I said, were critical to antiviral or Th1 immunity. And you'll notice I've stratified it again by IgG seronegative and IgG seropositive, is that we see a pretty good distinction between IL-2, but there's definitely some background levels, and noticeably in interferon gamma and TNF-alpha, that we find in our human subjects. And this corresponds, as I said, to the high levels of background that have recently been published by others looking at the SARS-CoV-2 T cell responses. And unfortunately, we have batch effects when we look at cytokines, which are really driven by the KIT standards that we use for cytokine detection. So we deal with this by normalizing fold change from untreated or mock treated cells. And that way we can kind of ignore any type of batch effect that we'll find. And if we know that this is driven by the cytokine standards, because if we use the same human sample in each, every time we run this, it's always, the fold change is similar, but the actual kind of quantity of cytokine responses differing. So now I'll show you data from 3 experiments combined of over 50 people stimulated in our assay. And you can see here's the level of anti-S IgG titer of those subjects. And here is the IL-2 cytokine secretion to, all of this is to the S peptide. Here's interferon gamma, TNF alpha, IL-5, and IL-6. And I hope you can see kind of very clearly the distinguishing feature is really IL-2 and interferon gamma, and not as much TNF alpha, IL-6, and IL-5. And if we look at that a little bit more closely at kind of a correlation matrix, you can see that both serum anti-S IgG seropositivity is what this variable means, or anti-S titer, are strongly related to the fold change of the IL-2 or interferon gamma. And that our values are, which, you know, as you get to 1, that means it's closer to a linear relationship, are starting to be much higher than what we saw with our activation assay. So this leads us to conclude that IL-2 and interferon gamma cytokine responses are strongly correlated to the anti-S antibody level that people are exhibiting post-infection. And when we look at whether these cytokine responses are related to actual kind of past history of COVID infection, such as the presence of fever or the loss of smell during infection, and it's not clear, it looks like there might be a trend if we get more subjects, as we continue to analyze more subjects, that IL-2 levels and interferon gamma levels are higher in people who experienced fever versus those who didn't. And similarly, we'll have to look at this here. I'm not sure this is gonna say anything for the loss of smell. So that's ongoing as we get more subjects. And then because we saw such a clear response for IL-2 and interferon gamma, I'm sorry, there's a typo here. In previous work that I'd done with tuberculosis vaccination, there was a whole body of literature talking about IL-2, TNF alpha, and interferon gamma as like those 3 cytokines really helping to drive potent immune responses and Th1 immunity. So I got interested in just looking at this data, combining those and seeing if a subject had this triple cytokines production. And that's another way to get rid of kind of that leaky background cytokine secretion that we're getting probably from cross-reactivity to other coronaviruses. And so this is just anti-S protein serostatus, so whether they have antibodies to the virus or not. And this is whether they, in the secretion assay, they had no or one of those cytokines being expressed at a greater than 2.5-fold than mock, or 2 of them is the double, or triple is all 3 of them. And I hope you can appreciate that it's really starts to become really specific that the people who are seroconvert also have this triple cytokine secretion in response to the S protein. And this also seems to drive the level of antibodies that we find because of based on the titer to the S protein being different between individuals who aren't secreting really any cytokines or their cells are secreting all 3 cytokines. And then of course, this leads us to some other interesting findings that we hope to get more numbers of is these people who have apparently high levels of cytokine secretion, but no antibody levels. So is this a T cell response in the absence of an antibody response? And then, you know, looking a little bit closer at those subjects, did they report multiple PCR positive events for which would indicate that they were infected multiple times or maybe had a longer lasting infection? So we're looking into that right now and hope to just get more individuals into those categories. And then kind of finally in conclusion, if you look at this kind of triple cytokine production, here's the IgG positive versus IgG negative to the different kind of antigens. You see it's pretty consistent between the different antigens that we're using to restimulate. but not always. So, sometimes you'll see a stronger response or weaker response between the M peptide and the N and S peptide. So it's mostly consistent, but as we get more people, it'll be interesting to see if we can pull out some other findings and relate that to either duration of immunity or antibody function. So in conclusion, in ongoing studies with healthy convalescent SARS-CoV-2 infected subjects, we can detect cellular immunity by activation assays or cytokine secretion, though background levels exist in our population, similar to previous reports. The magnitude of cellular activation is not linearly or strongly related to antibody levels, but perhaps it is related to antibody function. And we have collaborators on the NIH U54 project who are looking at antibody function that we're hoping to correlate our data to. And the magnitude of Th1 cytokine secretion, particularly the 3 cytokines that I talked about, IL-2, interferon gamma, and TNF-alpha, is strongly related to antibody levels. And we're hoping to do kind of some intracellular staining to confirm if this is from single cells that are producing all 3 cytokines, and if this is CD4 cells or CD8 cells. And that we think that some individuals, as reported by a few people, may have poor antibody responses, but evidence of T cell memory. And ongoing questions, we want to know, how does cellular immunity correlate to antibody function, isotypes, and neutralization? How do these change over time? How are these responses different in specific populations, such as kids, adults, cancer patients, pregnant moms? How does infection or vaccination only change these responses that I'm reporting to you here? And how does these kind of cellular memory responses that we're seeing help protect individuals long-term from SARS-CoV-2 strains or other coronaviruses? And I'll just say thank you to the people on my lab, Amelie, Ivy, Anissa, Io, and Sam, who worked on these studies. And then also the tremendous number of U54 collaborators at Tulane University. That project is led by James Robinson and involves just a really awesome team of individuals that none of this work would be possible without. And with that, I will take questions.
Thank you so much. I don't see any questions in the chat, but we can open it up to anyone in the audience who'd like to unmute themselves and ask a question.
Hi, Betsy. I have a question. How many total patients do you have in the study, or did you recruit?
So before vaccination started, I think the number is around 90 subjects, but it gets a little muddy because, we've been trying to get people before vaccination, and some of those people already have been infected. So yeah, I would say around 90 to 110.
And is everything done in real time, or is it frozen down and then thawed and stimulated and so forth?
We freeze it. Pros and cons of both. I've done the studies where you do everything real time, and it is kind of, I will say, nicer, especially if you can use lysed whole blood, because you use a lot less blood and you can get a lot more assays, and then, just do validation on the frozen PBMCs. But in our case, it's just logistically been too difficult to figure out how to do all of these collections and then test people, to avoid also the batch effects. We would just see so many batch effects when we do it, like weekly, right? For, I guess, like half a year.
Right. I mean, I was just wondering whether or not you stimulated in real time and then, froze the soups down, but you're actually freezing the blood down and then pulling it back out. Is that right?
Yeah.
Yeah. And there's seemingly
Which
a
means
lot
there's
of
no neutrophils, right? We have no neutrophils.
Right. Yeah. No, you're looking at PBMCs. There's a lot of people that are, seronegative. Is that, is surprising at this point? There's natural infected people, or what are the numbers of people who are IgG negative?
Yeah, well, so that's part of it is household contacts of people who were infected, and then also people who in the summer suspected they had been infected, but they would have been infected like in May or April or March when there wasn't a good time frame. Most of those people who were recruited, I was looking that up, in June and July had been infected 3 months prior. So they just didn't have the same type of testing then as
Right,
the people that we
okay.
were able to recruit into the study, you know, who had been infected over the summer or during the fall.
Okay. Yeah. So I guess you'll be able to do some different type of stratifications over time, since you don't know exactly who is COVID positive, but most of the,
We have, I would say we have about PCR results for, it looks like, for at least half of them to be 2/3. Kind of depends on where you divide the people for this infection time period before vaccination.
I guess, and you're right, the real interesting result will be to look at the vaccinated people with or without previous infection and see how that memory response or that, you know, occurs. I guess you can't do an actual CD8 cytotoxic assay with some of those cells.
So I would love to figure out a functional assay to do, but I was trying to figure out if there would be like, maybe we could do a pseudo assay one. And I'm hoping, because James Robinson is trying to get his pseudo assay up and running for antibody, that we could maybe potentially use that for a CD8 T cell killing.
Yeah,
I'm
that
not
would be
sure.
nice. That would be nice if you could do that from the CD8 side.
Yeah. I
Thank
think
you.
we do. I really just, I don't think we'll be able to do that until maybe like this summer, you know, get this first kind of analysis wrapped up. And like I said, the collaborators on that project have some really awesome kind of analyses going on with function of antibodies in terms of FC effector functions. So we're hoping to match some of our cytokine data with that and see if we, you know, other kind of interesting findings emerge.
Sounds good. Thanks.
Any last questions? Well, thank you so much. Thank you as well, Dr. Kracht, Dr. Norton, and all of the attendees here today. A recording of today's session will be available in the next 1 to 2 days. It will be shared via the LCATS listserv and posted to the YouTube channel. So please watch your email for notices about our next COVID-19 Chronicle Seminar. And we welcome more speakers for this seminar series. So if you or someone you know would like to share their COVID-19 related work, please be in touch. And thank you again. These presentations were both just wonderful. We really appreciate everyone's time, and just be in touch with any questions.